In this session, we are going to discuss about protein structure or structural organization of proteins. Proteins are made up of one or more polypeptide or protein chain. Proteins with a single polypeptide chains are called monomeric proteins, while the proteins with more than one monomeric units are called oligomeric proteins. Hence, some proteins may be made up of a single subunit or a single polypeptide. Some may be made up of more than one polypeptide chains. Hence, those which are made up of a single polypeptide, they are called as monomeric proteins. Those which are made up of more than one polypeptide chains, they are called as oligomeric proteins. Monomeric proteins are predominant in nature. They are in abundant quantity. If you come across second paragraph, each polypeptide chain is an oligomeric protein. In an oligomeric protein is called subunit or monomer. Consider if a protein contains three polypeptide chain, each of the polypeptide chain is called as a subunit. Example for oligomeric proteins which are made up of more than one polypeptide chain or hemoglobin because it is made up of four polypeptide chain, and lactic dehydrogenase which is made up of also four polypeptide chain. Each protein has a 3D structure which is referred to as its native conformation. In native form, proteins do not exist in a linear or a single dimension form. Instead they exist as coiled or folded structure or three-dimensional conformation. Only such 3D conformations are biologically active. The polypeptide chain which are obtained during the translation process from mRNA, that protein is crude and it will undergo post-translational modifications, and it will undergo various removal of certain amino acids and then forming a mature protein along with giving a particular 3D structure by undergoing folding, bending, twisting, coiling process that give rise to 3D structure of a protein and which is biologically active. This protein which has undergone coiling, bending, twisting, folding can be destroyed from its structure by denaturation, and that denatured protein is inactive. Only the protein which are in native form, they are biologically active. If you come across fourth paragraph, when the proteins are formed on ribosomes, they are synthesized in linear form. Formation of active 3D conformation from linear form of proteins is considered under the structural organization of proteins. So during the translation process, the linear form of proteins is undergoing structural modification to form a three-dimensional biologically active proteins. So let us see how proteins or structure is organized and how we are going to get information about the proteins. This structural organization of proteins can be exhibit under four different levels of structural organization: primary structure, secondary structure, tertiary structure, and quaternary structure. These four levels of organization will give you the information about a particular proteins regarding its number, nature, sequence, and type of foldings it is having, and all this information. Let us begin with the first one, that is primary structure. The primary structure of a protein indicates the number and sequence of amino acid residues from N terminal to the C terminal end of the protein. So the protein regarding information of how many amino acids are there, that is the number, and the sequence, that means the first amino acid, second, third, which is the amino acid present. The sequence is also informed in the primary structure from N terminal to C terminal. The features: peptide bonds maintain the primary structure. Peptide bond form the backbone of primary structure. Side chain protrude from the sides and in opposite direction. So the peptide bond is going to maintain the primary structure because this is the main bond which is involved in the primary structure. Later, whatever the structure you are studying, secondary, tertiary, quaternary, several other type of bonds are involved. In the primary structure, amino acids are arranged in linear chain. So second point, if you see, they are arranged in the linear chain. The folding, bending, twisting, coiling related information is obtained from the second structure, whereas primary structure is only a linear form of structure, and it also tells about number and sequence, and also the peptide bond is the primary bond in the structure. The primary structure of each protein has a unique amino acid sequence and numbers decided by genes contained in DNA, and primary structure is not affected during denaturation. So denaturation process is not going to affect the primary structure. If I want to show you in this diagram, here this is the diagram or illustration which shows the four level of organization: primary, secondary, tertiary and quaternary. So if I zoom the first one, that is primary protein structure, you can see the amino acids are there, that is in the circle they are amino acids, and in the bracket you can see sequence of chain of amino acids. Next is secondary protein structure. Here you can see hydrogen bonding of the peptide backbone causes the amino acids to fold into repeating pattern. They may be of two forms. You can see alpha helix and also pleated sheets. Next one is the tertiary protein structure. In the bracket you can see three-dimensional folding pattern of a protein due to side chain interactions. And then this whatever the secondary structure you have seen, that is alpha helix or pleated sheets, they'll undergo again folding, bending, twisting to form the tertiary protein structure. This for monomeric proteins, tertiary structure ends for monomeric proteins, whereas for quaternary protein structure can be seen in oligomeric proteins, where three to more than one polypeptide chain, that means two, three, or four polypeptide chains are involved. They are having their own tertiary structure for each of the subunits, and all these subunits combine together to form the quaternary structure. Protein consisting of more than one amino acid chain, you can see in the bracket in quaternary structure. So this is about four different level of organization illustration. So we are discussing in primary structure. If you see the importance of primary structure, the primary structure of proteins, specific sequence and number of amino acid is determined. The biological activities of the proteins are attributed to the three-dimensional structure. Therefore, the biological activities of proteins are in turn dependent on primary structure. So number and sequence of protein is explained in this heading. Coming to the secondary structure, if you see the secondary structure, where you can see alpha helix and beta pleated in this illustration that we are going to discuss now. Secondary structure refers to a primary folding of the polypeptide chain. The primary folding of the polypeptide chain, whatever you have studied primary structure, that will undergo folding. The alpha helix and beta pleated sheet structures are frequently found secondary structures of proteins. So usually they are frequently seen secondary structures in proteins. Beside these, some minor secondary structures like beta bend, non-repetitive secondary structures and super secondary structure are also present in the small amounts in proteins. So usually you can come across alpha helix and beta pleated sheet. The triple helix is usually seen in collagen, which is also called as collagen helix. So this alpha helix and beta pleated sheets that is in the second paragraph, you can see in this structure. This is the helix in the right side illustration, and the left side you can see the beta pleated sheet. The detail of alpha helix. Alpha helix is the most common secondary structure found in proteins. Alpha helix is most commonly seen in globular proteins, and alpha keratin as the exclusive alpha helix structure. So this is the alpha helix presence in proteins. Salient features of alpha helix: it is a right-handed coiled structure. Alpha helix structure is stabilized by extensive hydrogen bonding. Hydrogen bonding. Hydrogen bonds are formed between the peptide bonds that are three amino acid residues apart in the same chain. So the first amino acid, second and third amino acid. So that third amino acid combines with after the interval of third amino acid, uh, it will interact by hydrogen bonding to form a helix structure. For example, the peptide bond of the fifth amino acid is the hydrogen bonding with the peptide bond of the eighth amino acid. and 6th with the 9 amino acid. If you see the difference between 5th amino acid and 8th, it is the 3 amino acid intervals, and along with that 6th and 9th, here also you can see the 3 amino acid intervals. You can see the hydrogen bonding which can form helix structure. The hydrogen bonds are parallel to the helix. Alpha helix hydrogen bonds are individually weak, but collectively large number of hydrogen bonds maintain the helical structure. Even though hydrogen bond is weak individually, but collectively there are several number of hydrogen bonds which is going to make a stable structure of proteins. Each structure of helix contains 3.6 amino acid residues, and each amino acid is separated by a distance of 1.5 angstroms. So the pitch of the helix is 5.4 angstroms. So this is about the measurement of helix, the distance between the amino acids, and also distance between the uh, residues you can come across. Proline, hydroxyproline, glutamate, histidine, lysine, arginine, leucine, isoleucine, valine, glycine are considered as alpha helix destabilizing amino acid. Next is beta pleated sheet. So this is you can see in this structure, this is alpha helix. You are seen in the right side. Now in the left side, beta pleated sheet, that we are going to discuss now in detail. What is beta pleated sheet? The beta pleated structure is an extended structure. The beta pleated structure is stabilized by hydrogen bonding. Hydrogen bonds are between the peptide bonds of neighboring segments of the same polypeptide chain. Bonding chain lies side by side. The hydrogen bonds are perpendicular to the polypeptide chain. So the two polypeptide chains, they are going to bind, or they are going to bond by hydrogen bonding. And parallel and anti-parallel beta pleated sheets can be seen. In beta pleated we are having anti-parallel and parallel depending on the direction of the bonding of the chain. If you see the A parallel, if the bonding chains runs in the same direction, that means N terminal to C, and another is also from N terminal to C terminal in the same direction, then it is called as parallel sheet structure. Anti-parallel, if the bonding chain runs in the opposite direction, one is running from N terminal to C and another is running from C terminal to N terminal, that is seen in fibrin of silk. So this is about two different forms of beta pleated sheet structure, parallel and anti-parallel. You can see the parallel and anti-parallel sheets structure. Carbonic anhydrase shows both parallel and anti-parallel beta pleated structure, that is having both the structures. Next is the tertiary structure. If you want to study structure, let us see once again the figure of the tertiary structure. So this is tertiary structure which is obtained from the alpha helix and beta pleated sheets folding, bending and twisting. It will lead to tertiary structure. So tertiary structure of protein refers to the further folding of secondary structure. Further folding of secondary structure means what? What are the secondary structures? Alpha helix and beta pleated sheets. So they will undergo further folding. Polypeptide chain giving the compact three-dimensional conformation. In monomeric proteins, tertiary structure result in the final three-dimensional conformation of the polypeptide chain and formation of functionally active proteins. So the secondary structure will undergo folding, bending, twisting, coiling, forming the tertiary structure, which is going to form a functionally active protein. So proteins are active once it get into tertiary structure. Tertiary structure refers to the spatial relationship between the amino acid residues located far apart from each other in their linear primary structure but brought closer as a result of folding. So in this there is a further folding of secondary structure as you seen in the first line of the first paragraph. So while further folding and bending occurring, they will come closer to each other, and as a result there is a bonding takes place, and folding is going to be stabilized by various bondings. The tertiary structure result in the orientation of hydrophobic side chain towards the water free interior
and hydrophilic polar groups towards the surface of the proteins. So water loving amino acids will be exposed outside, and the water eating hydrophobic are oriented towards the interior part of the tertiary structure that makes the protein getting the particular type of tertiary structure. Bonds destabilizing tertiary structures. The tertiary structure is stabilized by weak non-covalent bonds like hydrogen bond, van der Waals forces, ionic bonds, and hydrophobic bonds. Even though these bonds are weak, a large number of these bonds give stability to the structure. So primary structure regarding peptide bond, secondary structure regarding hydrogen bond, whereas the tertiary structure we can see several of these bonds: hydrogen bond, van der Waals forces, ionic bonds, and hydrophobic bonds. Some proteins contain strong covalent disulfide bonds also, which are found between two cysteine residues, sulfur containing amino acid residues or cysteine residues of the same protein chain. Intra-chain disulfide bonds further enhance the stability of tertiary structure of proteins. This is our tertiary structure. Next is quaternary structure. If you want to go study quaternary structure, let us see once again the structure. This tertiary structure, it is for a monomeric protein. If the tertiary structure we are seeing like that, 3 to 4 subunits are there, they are going to combine to form quaternary structure. And how these quaternary structures are stabilized or organized, let us see in this heading, that is quaternary structure. Proteins having more than one polypeptide chain. More than one polypeptide chain means they are oligomeric in nature. In bracket you can see show one or more level of higher structure called quaternary structure. Quaternary structure refers to the spatial arrangement of the subunits of oligomeric proteins. So if more than one polypeptide chain is there, each of the polypeptide chain will having primary structure, secondary structure, and tertiary structure. So if you consider A, B, and C, the A, B, and C are three different polypeptide chain. A also is ending up to tertiary structure, B is also ending up to tertiary structure, C is also ending up to tertiary structure. So therefore we are having three subunits, that is A, B, and C. These A, B, and C come together. All the three will come together and they will make a group or join together. They will form a quaternary structure. So that is what the quaternary structure is all about. For example, instance, hemoglobin has four globin chains. Here you can see four globin chains, four subunits. So it shows quaternary structure. Other examples are aspartate transcarbamylase and lactate dehydrogenase. Like that, you can see come across several proteins with quaternary structures. Bond stabilizing quaternary structures. The quaternary structure is maintained by non-covalent bonds like hydrogen bond, van der Waals forces, ionic bonds, and also hydrophobic bonds, and also few covalent disulfide bonds. Quaternary structure have interchain. Inter means between, as I explained, A, B, and C, three different subunits. If the A and B is interacting, and also B and C, or C and A, or C and B, like that, it is between the chains. Hence the word interchain interaction between polypeptide chain takes place. Unlike tertiary structure which shows intrachain, within the chain there is a bonding. A is having its in its own chain, it is having a bonding. Within A there is a chain bonding, then it is called as intrachain. So between A and B, that is interchain. So quaternary structure shows interchain interactions. Importance of higher studies of proteins. Proteins are biologically active on only in their three-dimensional conformation. So the biological activities of proteins are attributed to higher structures. Up to quaternary structures, if they are in their 3D conformation, they are active. If a protein loses its three-dimensional form, that means quaternary structure or tertiary structure is going to lost. It loses its biological activity. You can destroy the whatever the secondary, tertiary, and quaternary structure of a protein by using some agents called as the denaturing agents, and the process is called as the denaturation. So if you eat it, or if you subject to chemicals or ultraviolet radiations and other factors, it leads to denaturation. So denaturation means the destruction of three-dimensional conformation of a protein, thus making it biologically inactive. Hence, that is our next topic, that is denaturation of protein. D means the first two letters DE. If you can come across destroy, destroy first two letter D, naturation, destroy the natural form of a protein. Denaturation. Definition: the process of disorganization of native protein structure is called denaturation. So the original structure is going to be destroyed by various process or factors or agents. Denaturation involves the loss of secondary, tertiary, and quaternary structure without breaking the primary structure. So the secondary, tertiary, and quaternary structure is going to be destroyed. Reason: higher structure of proteins are maintained by weak non-covalent bonds, as we just discussed now. Hydrogen bonding, van der Waals forces, ionic bonding, hydrophobic bondings, and they are going to be destroyed by the various factors and agents, which can easily disrupted by a variety of physical and chemical agents, whereas the primary structure of proteins are not easily broken because they are maintained by strong covalent bonds. Primary structure is having strong covalent bonds, whereas secondary, tertiary, quaternary are having weak bonding forces, hence they can be easily destroyed. Denaturation causes the unfolding of three-dimensional form of proteins. In this you can see in the diagram native form before the arrow mark, and after the arrow mark you can see denatured form. The in denaturation process, the native form is going to lose its three-dimensional conformation and becomes denatured. This process is called as denaturation. You can see above the arrow mark, and below the arrow mark you can write by using denaturing agents, which can be physical in nature or chemical in nature. So agent causing denaturation. So let us discuss which are the agents which destroy the natural form of the proteins, that is denaturation causing agents. Denaturation can be caused by various physical and chemical agents. Physical agents are pressure, heat, X-ray, UV radiation, and ultrasound. These physical agents can destroy the tertiary, secondary, and quaternary structure of proteins. And coming to the chemical agents, acids, alkali, organic solvents, ether, alcohol, higher concentration of urea, salicylates, and heavy metals like lead, mercury, and silver, they are also able to destroy the secondary, tertiary, and quaternary structure of proteins, leading to the denaturation and making them inactive. Biological relevance of denaturation: proteins are active only in their 3D form. Since 3D form is lost during denaturation, proteins lose their biological activity. Hence, if you are using a alcohol or a sanitizer where the alcohol is present, which is a chemical, it is going to denature the certain proteins in the microorganisms, which can cause diseases. Hence, this alcohol is very useful in destroying certain microorganisms. Denatured proteins are digested easily. This is due to the increased exposure of peptide bonds to proteolytic enzymes. For instance, cooking denatures dietary proteins. This is why cooked proteins is easily digested. So if you come across eating a meat without cooking and after cooking, without cooking it is difficult to digest, whereas the meat after cooking you can easily digest it, because the proteins will become loose by subjected to heat, because they will undergo denaturation, and that's make it easy to digest. So next we will discuss isolatory points of protein in the next session. Thank you.